Okay, so welcome to this next video in which we are discussing the macrolide antibiotics. So at the moment we are going for a revision of the process of translation in bacteria. Okay, so far what we have seen is that after assembling this 70S initiation complex, the next amino acyl tRNA, which has a complementary anticodon to this second codon here, is going to come in with the elongation factor TU with GTP bound to it. Okay, if the anticodon that came in was actually complementary to this codon that was in the second position, then the elongation factor TU will hydrolyze the GTP to GDP and inorganic phosphate, and then it will break off the amino acyl tRNA, and the amino acyl tRNA will be allowed to stay within the A site of the ribosome. Okay, so now what I wanted to discuss is just how these elongation factor TU proteins are recycled. Okay, so how do you reuse this elongation factor TU protein then once you have once you've cleaved it off the amino acyl tRNA and it's now bound to GDP? So what happens is another elongation factor comes along, and this time it's elongation factor TS. So this is EF for elongation factor, and then TS, okay? So what elongation factor TS is going to do is it's going to bind to elongation factor TU when elongation factor TU is bound to GDP, okay? And when elongation factor TS binds to elongation factor TU bound to GDP, the GDP falls off. Okay, so it's cleaved this GDP off already. Okay, and now simply what it does is it grabs amino acyl tRNAs and it grabs GTP. So it's going to grab a GTP molecule here. Okay, it's also going to grab an amino acyl tRNA, which I'll just denote like this. So a tRNA molecule with an amino acid bound to it at the end. So it's going to grab these two things, and it's going to bind them to the TU protein, so you get back amino acyl tRNA proteins with the elongation factor TU protein bound to GTP. So you regenerate these complexes, and then obviously the elongation factor TS comes off, so it breaks off at this point. So elongation factor TS then, a very important protein in recycling the elongation factor TU. Okay, right, so that's just how you recycle the elongation factor TU. Now let's turn our attention back to what we've just done. We have put in our second amino acyl tRNA into the A site. Okay, so this amino acid that's bound here, which is 4-methionine, is the first amino acid, and the amino acid bound here is the second amino acid. And you remember me telling you that the amino acids are bound to the tRNA by their carboxyl groups. So let me just remind you of the structure of an amino acid to make this crystal clear. Okay, so here's the structure of an amino acid. Here's the amino group. Here's the alpha carbon with a hydrogen coming off it. Here's the generic R group, so I won't put in a specific R group there, I'll just leave it general. And here's the carboxylic acid group. Okay, so this carboxylic acid group is bound to this tRNA in some way. Okay, so the amino group is free. Now, what is going to happen basically is that the enzyme peptide transferase, which is here, let's say, which we'll colour in a more substantial colour than yellow this time, we'll colour it in orange. So this is the peptide transferase portion of the 50S ribosome, which is going to catalyse the transferring of the amino acid on this amino acyl tRNA onto this amino acyl tRNA, basically. This is the enzyme peptide transferase. I should write its name out here. So this is peptide transferase. And basically what it's going to do is it's going to cut the amino acid that is bound to the amino acyl tRNA in the P site off. Okay, so it's going to regenerate its carboxylic acid group, and then it's going to bind it to the amino group of the amino acid that is bound to the aminoacyl tRNA of, is bound to the tRNA in the A site, basically. That's what that's what peptidyl transferase is going to do. So, I'll say that again because it's worth repeating. It's going to cut this amino acid off here, okay? It's going to cut that off, and that means that it's going to regenerate this carboxylic acid group. It's then going to bind this amino acid to the amino group of the amino acid that is bound to the tRNA in the A site, which we know is exposed because this amino acid is bound via its carboxylic acid group. So, what's going to overall happen then, if we draw the next stage? So here is the 30S ribosomal subunit here. Okay, here's the mRNA in the middle here. Here's the 50S ribosomal subunit here. Okay, and now we still have this tRNA here, but no longer does it have the 4-methionine bound to it. Instead, you now have the second tRNA, which has not one amino acid anymore, but 2 amino acids bound to it. Okay, so this amino acid here is the 4-methionine tRNA in blue, and in green, this is the tRNA. This is the amino acid that's was originally bound to this tRNA in the second. In the A site. So this is the A site, this is the P site, and then there will be the E site, which nothing's in at the moment. Okay, so here is still the start codon in turquoise. Here in orange is the second codon of the mRNA. Then in purple, here is the 30S ribosomal subunit. Okay. And then in light green is the 50S ribosomal subunit. So peptidyl transferase overall catalyzes the movement of amino acids from the tRNA in the P site onto the tRNA in the A site. Okay, so it forms the peptide links between the amino acids. So you now have a peptide chain consisting of 2 amino acids that is held onto this tRNA that's in the A site. Okay, what now has to happen is the mRNA basically has to move through, or the ribosome has to move along the mRNA. In fact, it's actually a case of the ribosome moving along the mRNA more than the mRNA being moved through the ribosome. But basically what has to happen is there has to be a relative movement basically. You need the start codon, i.e. the codon that's currently in position P, to move into the E site. You need the codon that's currently in position A to move into the P site, and you need the next codon along here, so let's colour it in red. You need this next codon along here to move into the A site so that you can then bring in another aminoacyl tRNA, which is complementary to this third codon here. Okay, so for this process to happen, for this movement to happen, what has to happen is you need an elongation factor, another elongation factor known as elongation factor G. So G here stands for elongation factor EFG, okay? And it has GTP again bound to it. So here is elongation factor G with GTP bound to it. It is going to come in, bind to the ribosome, cause it to move along the mRNA, and where does the energy that you need in order to move the ribosome come from? Well, it comes from the hydrolysis of the GTP. So you get GDP and inorganic phosphate. So the elongation factor becomes bound to GDP and inorganic phosphate. That hydrolysis of GTP releases energy that is used to move the ribosome along the mRNA. Okay, now you might ask, how does the elongation factor G get recycled? Well, it's going to cleave off from that GDP, so you'll get elongation factor G, and then it will recombine with another GTP molecule simply to make the GTP back again. Okay, so that recycling process is very simple. Okay, so overall what this is going to result in is, let's draw this next step, is here's the 30S ribosomal subunit here. Okay, here's the mRNA now. Okay, and here is the 50S ribosomal subunit. And now this original tRNA that was in the P site is now in the E site. The tRNA that was in the A site is now in the P site with these 2 amino acids off it. And now there is a new vacancy in the A site, basically. So here now is the start codon in turquoise. Here is the second codon of the mRNA in the P site. Okay, and in light green we have the 50S ribosomal subunit here. Okay, and then in purple we have the 30S ribosomal subunit here. Okay, right, so now what's going to happen is you're going to start the whole process of elongation again. So what will happen is another amino acyl tRNA will come in here. Again, it will have the elongation factor TU bound to it, and that will check that the amino acyl tRNA that comes in and binds to this third codon here actually has a complementary anticodon to that third codon. Then what will happen is, once that process is successful, the peptidyltransferase enzyme, which is somewhere down here, okay, will transfer both of these amino acids now. So remember, what you have is this first amino acid, this 4-methionine, bound to this second amino acid by an amide link. The second amino acid is then bound to the tRNA by its carboxyl tail. You will cut that link there, this link here, and you will bind this dipeptide onto the third amino acid that is going to come into this A site, basically here. Okay, so here comes in this third amino acyl tRNA there. Okay, and you will make a tripeptide. In addition, this tRNA in the E site, that will leave, basically. That's why this is called the E site, for the exit site, basically. Okay, so you will then have a tripeptide in the A site bound to this tRNA. The elongation, the movement process, sorry, will happen again, and you will move this amino acyl tRNA, well, now with a tripeptide on it, into the P site, and the elongation process will continue. Now, what will gradually happen is you're going to build up a longer and longer polypeptide attached to this amino acid in either the A or the P site, basically. Well, it will be transiently in the A site before you move, and then it will be in the P site. Okay? So you're going to build up a longer and longer polypeptide. Now that cannot stay within the ribosome. So what you have basically on the 50S ribosomal subunit is a hole, basically. Here is this hole. I'll highlight it in red. And as the polypeptide grows, what's going to happen is it's going to be fed out of this hole into the cytoplasm. So this hole here is known as the polypeptide export tunnel. So this is the polypeptide export tunnel. And in order for translation to work, this tunnel has to be viable, basically. You have to be able to feed the growing polypeptide out of the polypeptide export tunnel. This polypeptide export tunnel is the target for the macrolide antibiotics. Okay, so the macrolide antibiotics, what they do is they bind to this 50S ribosomal subunit, and they block that tunnel. So they bind to this portion here, and they bind over the polypeptide export tunnel, blocking it. So that means that this growing polypeptide can't get out of the ribosome, so it gets stuck within the ribosome. It's what one of my teachers once used to call ribosomal constipation, basically, molecular constipation. So the polypeptide gets bunged up inside the ribosome, can't get out, and eventually you can't continue. You'll get to a point where you can't continue, and that will terminate translation. Okay, so it stops protein synthesis in that way. So I will conclude this talk by giving you some examples of macrolide antibiotics. So they generally have the suffix phromycin. So erythromycin is the archetypal example of a macrolide antibiotic. It's very safe generally. It's antibiotic that is commonly prescribed by GPs. Okay, another example is clarithromycin. Clarithromycin. Okay, again, it's an antibiotic that is commonly prescribed. Then we get to more rarely prescribed ones. So I'll continue up here, I think. So another one is roxithromycin. Roxithromycin. Okay. Then azithromycin. So azithromycin is another example. These are extended spectrum macrolide antibiotics. Azithromycin. And now we're really getting more dangerous. Telithromycin. Telithromycin has a black box warning in a box warning in America, which basically warns about how toxic it is. So it's a nasty drug that you would only be prescribed if you had a very serious, life-threatening bacterial infection. Telithromycin. And those are the 5 examples that I can get that are given in humans. There is also a very important example used in veterinary medicine, but not in human medicine, known as tylosin. Tylosin is used a lot in veterinary medicine. So it's an important example, but it's not really used in human medicine, so I'll just put that. This is used in animals, animal medicine, veterinary medicine. So if I've got any vets watching this, that's for you.